Hi, my name is Vitoria, and I'm going to present my work entitled Cell Biology and Bioinformatic Mapping of Gene Regulatory Networks in Sensitive Neuron Development. So, the dorsal root ganglia is one of the components of the peripheral nervous system, and anatomically he is located bilaterally to the spinal cord, as you can see here in this image. And in the interior of this structure, the cell bodies of the sensitive neurons are located. And those sensitive neurons can be classified in three subtypes: the proprioceptive neurons that will respond to temperature stimuli, the mechanoceptive neurons that will respond to mechanical shocks, and the nociceptive neurons that will respond to pain stimuli. So the differentiation of those three subtypes of sensitive neurons in the DRG is dependent of the expression of specific genes. So we can see here in this image the neurocrust cell migration. After that, those cells will commit to a sensory lineage fate, and with the expression of specific transcription factors, those cells will differentiate into the sensitive neurons, the nociceptive, the mechanoceptive, or the proprioceptive neurons. So the question here is, which transcription factors act in this transition between sensory lineage and differentiated sensitive neurons? So the SCRATch2 is a neurotranscription factor. He was described as a transcription factor that is expressed in the nervous system, as we can see here in this image. And our question here is, what is the position of this neurotranscription factor, SCRATch2, in this cascade that will result in the differentiation of the sensitive neurons? So, considering that SCRATch2 is a neurotranscription factor, and also that SCRATch2 binds to the DNA, to the genome, recognizing sequences called SASBOX to regulate the activity of other genes, we asked two questions. The first question, what genes are co-expressed with SCRATch2 in the dorsal root ganglia? So we answered that question using immunofluorescence and in situ hybridization. After that, we asked which are the possible target genes of SCRATch2, so we used open source bioinformatic tools to answer that question. With the analysis of in situ hybridization and immunofluorescence, what we saw was that SCRATch2 here in this picture have the expression pattern similar to the expression pattern of other differentiation markers as HUCD and islet1. And also we can see a complementary pattern of expression between neurogenin2 and SCRATch2. So that can indicate that SCRATch2 possibly acts in the differentiation phase of those sensitive neurons. So to see if SCRATch2 regulates the activity of islet1 and neurogenin2, we search for regulatory regions of islet1 and neurogenin2 using UCSC genome browser, and after that we asked if SCRATch2 recognizes any of those regions. So we use UCSC genome browser, tracks of cut and run for SCRATch2, and we also searched for SCRATch2 target sites. So what we saw was that for islet1 we found 7 regions that SCRATch2 recognizes, 4 intergenic and 3 intronic regions, and of those 7 regions, 3 presented SASBOX sites for SCRATch2. For neurogenin2, we found 3 intergenic regions that SCRATch2 recognizes, and in all those 3 regions we found SASBOX sites for SCRATch2. So that indicates a possible regulation of islet1 and neurogenin2 by SCRATch2. So to finish this presentation, I would like to thank the members of the Laboratory of Vertebrate Molecular Embryology in the University of São Paulo, and also the funding provided by FAPESP and CAPES.